Hello, today we are discussed in topic glycolysis. The process of glycolysis was discovered by three scientists, named as Emden, Meyerhoff, and Parnas, so it is also called as the AMP pathway. Glycolysis occurs in both aerobic and anaerobic organism. But the pathway is itself anaerobic, that means no oxygen required in this process. All the reaction that involved in glycolysis takes place in cytoplasm of cell. Sequential oxidation of glucose into two molecules of pyruvic acid is simply called as glycolysis. Glycolysis is mainly divided into two phases. The first one is energy requiring phase. In the second is energy releasing phase. In energy requiring phase, ATP are used up. In step 1, one molecule of glucose is converted into one molecule of glucose 6-phosphate. An enzyme used in this reaction is hexokinase. Hexo means 6 and kinase is used for phosphorylation. In this process, one ATP is used up and it converted into ADP. Magnesium ions are also used in this reaction as catalyst. In step 2, one molecule of glucose 6-phosphate is converted into one molecule of fructose 6-phosphate, which is a reversible reaction. An enzyme used in this reaction is phosphohexoisomerase. In step 3, one molecule of fructose 6-phosphate is converted into one molecule of fructose 1,6-bisphosphate. An enzyme used in this reaction is phosphofructokinase. One molecule of ATP also used in this reaction, and ATP converted into ADP by giving one phosphate molecule. Magnesium ions also used as catalyst. In step 4, one molecule of fructose 1,6-bisphosphate is converted into one molecule of phosphoglycerol, which is commonly called as 3-PGA, and it is 3-carbon compound. And also one molecule of dihydroxyacetone phosphate, which is commonly called as DHAP, and it is also 3-carbon compound. Enzyme used in this reaction is aldolase. In step 5, dihydroxyacetone phosphate is converted into 3-phosphoglyceraldehyde. For further reaction, this reaction interconvertible. Enzyme used for this reaction is phosphotriose isomerase. In energy releasing phase, ATP are formed. In step 6, two molecules of 3-phosphoglyceraldehyde is converted into two molecules of 1,3-bisphosphoglyceric acid, which is reversible reaction. Enzyme used in this reaction is phosphoglyceraldehyde dehydrogenase. Additional phosphate come in 1,3-bisphosphoglyceric acid is from H3PO4. In this step, aldehyde is converted into carboxylic acid, which is oxidation reaction, which drives the formation of acyl phosphate bond. In this step, an AD is converted into an ADH2. An ADH2 is source of ATP. It generates ATP when it enters an electron transport chain. In step 7, two molecule of 1,3-bisphosphoglyceric acid is converted into two molecules of 3-phosphoglyceric acid, which is reversible reaction. An enzyme used in this reaction is phosphoglycerokinase. Additional phosphate loss from reactant is taken by ADP and converted into ATP. The ATP direct formed in form of substrate is called as substrate level phosphorylation. Magnesium ion are also used as catalyst. In step 8, two molecules of 3-phosphoglyceric acid is converted into two molecules of 2-phosphoglyceric acid. which is reversible reaction. An enzyme used in this reaction is phosphoglycerate mutase. In step 9, two molecules of 2-phosphoglyceric acid is converted into 2 molecule of phosphoenol pyruvate, which is commonly called as PEP. It is reversible reaction. An enzyme used is enolase. 2 water molecules are losses in this reaction, and magnesium ions also used. In step 10, 2 molecules of phosphoenol pyruvate is converted into 2 molecule of pyruvic acid. Enzyme used in this reaction is pyruvate kinase. In this reaction, 2 ADP molecules are converted into 2 ATP by taking phosphate from PEP. In step 10 and step 7, direct ATP is formed, so it is also called as substrate-level phosphorylation. Gross product of glycolysis. To pyruvic acid, 2 molecules of an ADH2, which gives 6 ATP, when enter an electron transport chain, for ATP and substrate level. Gross ATP in glycolysis is 10. Net product of glycolysis. Number 1, 2 pyruvate. Number 2, to NADH2. Number 3, 2 ATP. 2 ATP used in energy requiring phase. Net ATP of glycolysis is 8. Thanks for watching. If you liked the video, please subscribe my channel for more educational video.